So this is going to be a quick video on how to use the dashboard to understand what's going on with the results of your metagenomic data analysis. So we're on the T-BioInfo platform. We kind of process this data either with KIME, CHIME, or with Data2 pipelines. And here we have kind of the basic composition of our data. So we can select here level of taxonomy. For example, we can go higher on the level of taxonomy, or we could go lower on the level of taxonomy. Remember that a lot of these are not going to be mapped well. So for example, you can see that the majority of the data at the species level is ambiguous. So if we kind of look at this, it tells us right away that maybe that's not a very good way to look at that. Actually, I see also other. Okay, there we go. So a lot of this taxa is not going to be mapped. So you can remove that simply by clicking on that particular taxa. So here now you can hover over and you can see what are these taxa. And a lot of times these are unidentified species, uncultured, right? You can see there's a lot of issues at this lower level. So let's go to the phylum level. So here it's very clear. The majority of them are annotated, so we can remove the other, and we can see that there's some variation. So let's say we kind of spot visually that there's a difference between these two samples. So let's take a look, for example, at something like, let's just go through. For example, if you double click, you will only get that particular taxa. So here we can go through and see the differences. And it looks like there might be some differences between the two groups. So here we have the control, and here we have the psoriasis. Let's go and maybe take a look at another one. For example, chloroflexi is only present in psoriasis, but only in 2 samples. So it's not a lot of confidence that this is actually significant, right? Cyanobacteria seems to be present abundant both in control and psoriasis. Here again, only 1 sample. So we can kind of go through. Now, a lot of times that's the limitation of, again, this taxonomic level. It doesn't provide us with a very good resolution, and a lot of it seems to be kind of all over the place. And the main reason for that is because there's so many actual bacteria that fall under these phyla. So let's go into the order. Here again, we're kind of visually, it might be a little bit confusing. So we might want to take a look at some of these examples. Maybe, for example, this one is only present in controls. Okay, and so just by exploring our data, we can start identifying some trends. And these trends could be very useful because we can start asking specific questions about these types of bacteria, and we can also manually find some of them that are very interesting. So here, for example, okay, here we have some bacteria that's maybe more abundant in the control. Or this bacteria is more abundant maybe in the psoriasis. So that's how you can navigate this. Now, in addition to this, you have here some kind of a global comparison between the different groups. And a lot of times this depends on the annotation. So for example, when you upload your metadata, you can have multiple types of annotation. Sample name, for example, here we have control and psoriasis. And here we have control and psoriasis, but we can change this. If we had kind of other types of information, we could definitely look at that. Now, some of this information is not very useful. Let's take a look at a different example. This different example is going to be with a little bit more metadata. So here you can see a much clearer example. This is a comparison between BV and normal, but you have quite a number of different types of phenotypes added to the table. So this is specifically vaginal microbiome. Now it's very clear, right? You can see right here, lactobacillia. So we can find it right here. And you can see that it is much more prevalent for normal and less abundant for BV. Now here we can also change what exactly are we looking for. For example, gestational age, or we can look, for example, by race. So here we're looking by race. There's black and white. And I don't know if we necessarily expect there to be a lot of variation, but we can certainly check. And there could be some variation. So we can identify manually some of them. For example, you can see all of these. They're not very consistent across this group, but they're definitely more present for white women rather than black. Now you can do the same thing over here. You can, for example, this is term and preterm birth, and you have phenotype. You have disease right here. Maybe we can change this to disease as well. So you can definitely play around with the data and kind of understand global differences, individual differences at different levels of taxonomy, and also alpha diversity plots. Here we're by race. Let's take a look by phenotype. We have term versus preterm birth, and we also have disease. So we see BV versus normal. This seems to be less number of OTUs in this group. So this dashboard can give you insights and can give you some ideas on how to perform downstream analysis. Down here you will find the actual table. You can download it and export it by the different level of taxonomy as well. And if there's an image that you would like to save, for example, you saw that maybe maybe this is a group or this is a taxa that you want to highlight, you can download, either you can open the HTML or an SVG, and so that will download a figure that you can then open up, and you can see right here. We can preview, or you can even open it in Adobe Illustrator to edit it. And you can see that this is something that is easy to work with, and you kind of have all of the annotation right here. Or you can also download this as an HTML, and that HTML is something that you can now open a bigger view of the same figure. And that can help you kind of, make it more, stretch it and kind of save it the way that you would like. Over here, you can actually now create a PNG, and that's a plotly function. So this would be a pretty high quality figure that you can include in your presentation.